Hello, gentlemen. This photosynthesis lab that we've just performed is a tricky one. In fact, it's actually an AP biology lab. It's tricky. Not only is it tricky to perform the actual lab, it's also tricky to look at the data, analyze it, and understand what it's trying to tell you. So as a result, I'm going to use this recorded lecture to walk you through the graphing of the data graphing, as well as what this graphing means, or what the data is trying to tell us, which is to say the analysis. So what I'd like you to have out in front of you is a piece of paper and a pen or a pencil, as well as your data that you gathered in class on Friday. First of all, let's just recap what we did in lab, what we were trying to test, and what we were looking for. We were testing, as you can see, the effect of either CO2 concentration or light intensity, one or the other, on the speed, on the rate of photosynthesis. And we can determine that speed or that rate by how fast our leaf discs float. A faster time, a shorter time, means that photosynthesis is happening faster, and a slower time for leaves to float means that photosynthesis is happening slower. So our goal in this lecture, like I said, is to actually determine exactly how fast the leaves generally floated in each cup. And we do that by analyzing. And we're going to analyze our data. But how are we going to analyze it? Well, first we have to graph it so that we can analyze it. So let's move forward. Before we go on, though, let's just talk about a few guidelines. Number one, I'd like you to use your data, your own data. Now, I'm going to be giving you some sample data, but I'd like you, if you can, if you have good data, to use your own data and to graph your own data. So whatever I do here in this lecture is just guidelines and a guide and suggestion as to how to work with your data. Now, if you don't have good data, if your data is no good, then you can borrow mine. If you have no good data for your high CO2 or light intensity data, then borrow the high data. If you have no good data for the low CO2 concentration or light intensity data, then you can borrow the low data. If you have poor data for both high and low, then borrow both of my high and low. But again, I stress that you actually use your own data. Well, what is good data? Well, good data means that your high, either concentration or intensity cup, has 5 or more leaves float. So if you had 6 or 7 or 8 or 9 leaves float in the time that you recorded data, that's good data. If you only had 2 or 3 or 1 or no leaves floating, then that's no good. That's not good data, and so I want you then to use the data that I will be presenting you now. Alright, so now let's actually move forward. Alright, this page looks something like you should have in your handout. You should have a data table with some numbers filled out. You have 3 columns on that data table. You have the time in minutes. You have the number of leaves floating in either high concentration or high light intensity. And you have the number of leaves floating in low CO2 concentration or low light intensity. Alright, and so you can see a list of numbers going from 1 to 15. And according to each of those numbers, we have a number of leaves floating. Now, what we'd like to do with this data is to begin to graph it. Now, you are actually in your lab report going to have 3 separate graphs. 2 of them are going to come straight from this data. Okay, so you're going to have 2 graphs. The first graph, the x-axis will be time in minutes, and the y-axis will be number of leaves floating in the high concentration or high light intensity. The second graph that you have will also have time as your x-axis, and it will have the number of leaves floating in low concentration or low intensity as your y-axis. As you can see, sort of with your naked eye at this data, the 2 columns of leaves floating you can tell there's a difference. Certainly, in the high concentration, we had leaves floating sooner. We had leaves floating at minute 4, where the first leaf that floated in the low intensity was at minute 6. And then you look at the very bottom, the very end of our experiment, we had all 10 leaves floating in this hypothetical sample data. While in the low concentration or low light intensity, we only got 4 to float in the 15 minutes of data collection. So again, let's take a look now at how we will be graphing these. Let's now graph the high concentration of CO2. Or if you're working with light intensity, that's what you're going to be graphing here. All right? So this could be either your high concentration or high intensity. Now notice how I've titled this graph. I'd like you to title your graph. Notice how I've labeled the axes. All right? The y-axis, here's our y-axis, here's our x-axis. Our y-axis is labeled leaves, and our unit of measurement is number. So every dash or every hash mark is the number of leaves. And our x-axis is labeled as well with time, and the unit of measurement is minutes. Right. Scaling our axes now is pretty easy. Right. For our high concentration, if you look back at the data, our high concentration, at the end of 15 minutes, we had all 10 leaves floating. So the top of our y-axis will be 10. And now we can simply scale by ones all the way down to 1. So we go 9, 8, 7, 6, 5, 4, 3, 2, and 1. And we can start labeling those. All right, you get the picture here. I'm not going to label each hash mark. Labeling the x-axis is just as easy. We started with minute 1 and we went to minute 15. Let's label our axis there. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, and I'm running out of room, 15. I'd like you to title your axes with units of measurement and then scale and label the axes. Can you do that now? After you've accomplished the work on the axes, now let's start plotting our data. We'll plot in blue. All right. If we look back at our data, we can notice that the first 3 minutes of this experiment, no leaves were floating. And so we're going to plot a 0 on minute 1, on minute 2, and on minute 3. At minute 4, we saw 1 leaf floating, so you're going to plot a 1 at minute 4. The same was true, there was only 1 leaf floating at minute 5. At minute 6, we saw that there were now 2 leaves floating, and so you'll plot minute 6. Alright, and you'll continue to do this for the rest of your data up until minute 15. Please do that now. Alright, I've plotted the rest of our data, and as you can see, over time we see more and more leaves floating, until at the minute 15 we finally saw all 10 leaves floating. Now you should look and see something that looks something like this on your graph with a bunch of dots. Now our job is to connect those dots. We'll connect them with a black line. Okay. And so again, if those dots are on the x-axis, there are no leaves floating, we will just connect them along the x-axis. Then we will continue moving up and over and up and up and up, over, and continue the rest of the way. And this is what our graph looks like. Now again, this is our high concentration of CO2 or our high light intensity. I'd like you to make the same kind of graph with the same axes, the same units, and the same scale, but a different title and different data using your low concentration or your low light intensity data. Please do that now. Now, here is a similar graph that I've created with regards to our sample low concentration of CO2 data, or you can use this as, again, your low light intensity data. Now again, I will be connecting the dots along the x-axis, connecting them as leaves start floating at minute 6 and on, and then here we go. Here's our data. Your graph should look something like this. Maybe not exact, but something similar. Now, here's where it gets a little bit more complicated. In a simpler lab, or a lab that's not really meant or aimed for an AP class, we would simply take the average time it took for leaves to float. However, as some of you realize, especially for our low concentration or low light intensity, some of our leaves didn't float. In many ways, you could say that it took an infinite amount of time for them to float. So taking an average wouldn't be very useful because the infinity kind of leaves would really skew our average. So instead of taking an average, we're going to use another kind of value that gives us a general indication as to when leaves started floating. That value is known as ET50. ET50 stands for the estimated time, ET, that it takes for 50% of the leaves to float. So for about half of the leaves, so if half of the leaves are floating, that gives you an indication as to when generally the majority of leaves started floating. Now we have to ask ourselves the question, what's 50% of 10? Because most of us use 10 leaves. Well, that's a pretty simple question. 50% of 10 is 5. So now we have to determine in each of our cups, when did 5 leaves start floating? And now we can take a look back at the graphs that we calculated. All right, now here we are back at our high intensity or high concentration graph. And let's take a look and see how or when 5 leaves began to float in this cup. Well, here's our 5. We'll circle the 5. And we'll draw a straight horizontal line from the 5 on the y-axis straight towards our line, or our graph. And now, keep in mind, my graph isn't perfect, but this is my straight line straight to my graph. And I've hit my graph at this point. And once you hit your graph, stop. Stop drawing your line. Instead, now I'd like you to draw a line from that point straight down, vertically down. And it should cross the x-axis somewhere. Now again, my graph isn't perfect, but it does cross my x-axis at around 8 minutes. So, what that tells me is that when I interpolate from 5 leaves, I hit 8 minutes, which tells you that my ET50 value is 8 minutes. I want you now to determine your ET50 value. Draw it on or write it on your graph and then circle it, please. Now when we look at the low concentration or low light intensity graph, we notice something interesting. When we look at 50% of the leaves we used here, which again is 5, and we draw a straight line, we don't hit our graph. Now keep in mind that's not perfectly straight, but either way, we would never hit our graph because if you look at the data, 5 leaves never float, not at least within the 15 minutes that we recorded. So what that tells us was when we create our vertical line, our ET50 value is greater than 15 minutes. Okay, and so that's if your leaves or if your data says that less than or fewer than 5 leaves ended up floating, that means your ET50 value is greater than 15 minutes. It took a very long time for half of the leaves to start floating. This is going to become important when we make our third graph. I'd like you to now your second ET50 value. Now this is where it might get really confusing, so pay close attention here. Remember that this lab is sort of like the lab that when we studied enzymes. It was sort of like our enzyme lab in that we used hole punches of things and we timed or measured the time it took for that hole punch to float. And just like the enzyme lab, we were measuring some kind of activity, whether it was enzymatic activity or photosynthetic activity. And with any kind of molecular or biological activity that we're timing, remember that when we had high enzymatic activity, the time was very low. And when there was low enzymatic activity, the time was very high. So this is low activity, and this is high activity. Now, if you just took a glance at this graph, you'd see low activity, well, doesn't that look like it's really small? And high activity looks like it's really small. It's not an intuitive way of depicting enzymatic activity. And so what I had actually asked you to do in our enzyme lab is not to take time, but rather to take 1 divided by time. So let's do that. I'm going to erase all of my red, and I'm going to erase all of my black, and I'm going to change my y-axis into 1 divided by time. Or we can just call it 1 over time. It means the same thing. And when we graphed 1 divided by time, what we found was that 1 divided by a really big number is a very small number. And when we divide 1 by a small number, it's a relatively bigger number. And so now what we see if we graph a bar graph is a more intuitive sense, or a more intuitive picture of activity. High activity is the bigger bar, and low activity is the smaller bar. Now, of course, this is the y-axis is now a small decimal, but we can more intuitively look at the graph and understand the intensity of enzymatic activity based on enzyme concentration. We're going to do the same thing here for our photosynthetic activity. Now, keeping in mind that ET50 is just a measurement of time, so in many ways we're doing the same thing. So let's take a look at that. So what we have here is a third graph. Let's call it graph number 3. And I've titled it as the effect of CO2 concentration on the speed of photosynthesis, or the magnitude of photosynthesis. And so our x-axis will be the CO2 concentration, and our y-axis will be the speed. Oops, that's a y. That should be a y. All right, let's cross that out. Our y-axis will be our speed of photosynthesis. Well, our speed or magnitude of photosynthesis will be indicated by our 1 over ET50. So that's our y-axis, which is the speed of photosynthesis. All right, and our x-axis is our CO2 concentration. And we're simply going to have two values here. We're going to have our low CO2 concentration and our high CO2 concentration. And now we have to figure out how to scale our y-axis. Right? Well, our y-axis is our 1 over ET50 value. So now we have to take a look back at our ET50 values. So let's take a look at our ET50 values. Our high concentration ET50 value, remember, was a value of 8 minutes. Okay, and so we have to ask ourselves, what's 1 over 8, or 1 divided by 8? Using your calculator, you'll see that it's 0.125, or we can round it off to 0.13. And our low ET50 value was a number that was greater than 15. Well, let's use a number that's greater than 15. Let's use 16. So let's say our ET50 value is 16 minutes. So our 1 divided by ET50 value will be 1 over 16. Well, if you use your calculator there, it will be 0.0625, but we'll round it off to 0.06. Now let's evaluate which of these numbers is bigger, because that's going to be the top of our y-axis. Thinking carefully, our bigger value is 0.13. Right? That will be the highest point on our y-axis, and that's how we'll scale our y-axis. So let's take a look at that graph again. Here we are back at our graph, and we will put the top of our y-axis as a value of 0.13. And we'll treat this going down as almost going by ones. So we'll go 0.12, 0.11, 0.1, 0.09, 0.8, 0.6, 7, 0.6, 0.5, 0.4, 0.3, 2, and 1. Alright, and I'll label these in a second. There, I've labeled them. So now we can graph our 1 over ET50 values. Okay, so looking at our low value, remember that our 1 over ET50 value was 1 over 16. Well, 1 over 16 is 0.06. Let's put a dot there, and let's turn that into a bar graph, or a bar. And our high concentration was at the very top because 1 over 8 was 0.13, thereabouts. And we'll put a dot there, and a bar. And there's our bar graph. And there's our third graph. And hopefully we can now draw conclusions from this graph. We can ask ourselves, what effect does CO2 concentration have on the speed of photosynthesis? Well, I hope that this graph now shows you that the speed of photosynthesis is great when CO2 concentration is high. And when CO2 concentration is low, the speed of photosynthesis is not so great. It's much lower. Okay, and so that can help you draw some conclusions. And now we have to ask ourselves the question, why is this true? And maybe looking at the equation for photosynthesis, we can get some answers. We can get some answers because, hey, look, CO2 is one of the reactants. It's one of the ingredients in the photosynthesis equation. All right? In the whole process of photosynthesis, you need to add CO2. And hopefully, and when you do add more CO2, you get more products, and you get them more quickly. Just like if you have more flour, you can make more bread more quickly. Or if you have more sugar or eggs or butter, you can make more cookies or cakes more quickly. It's just like a recipe. It's just like a recipe for some food or a recipe to make sugar and oxygen. That's what photosynthesis is. And so now we can start drawing conclusions, and hopefully, now that we have our three graphs, we will include those graphs in our lab reports, and we will now be able to begin writing the introduction and begin drawing conclusions in that lab report. Now, if you have any questions, please don't hesitate to email me over the weekend. This lab report, remember, is due on Monday. All right, so if you do have questions, please contact me. Good luck, gentlemen, and I'll talk to you later. See you Monday.